The first item that I talked about was stereotaxic surgery. So stereotaxic surgery is a technique that allows researchers to place a cannula or an electrode into deep and small structures within the brain. And this surgery technique makes it possible to stimulate and record wanted structures through the use of electrode without disturbing underlying or surrounding structures. Researchers that use stereotaxic surgery also use antiseptic techniques, such as wearing surgical masks or gloves, in order to keep the field sterile and safe for the animals that are undergoing surgery. These animals will also be monitored for 1 to 2 weeks after surgery to make sure that the surgery is not negatively affecting them. The second item that I talked about is the difference between gray and white matter. So gray matter can be found in the darker areas of the brain, and white matter can be found in the lighter areas of the brain. Gray matter surrounds areas such as the cerebrum, and that is known as the cortex. And white matter can be found inside in subcortical structures in areas such as the corpus callosum. Gray matter is made of neural cell bodies and their dendrites, and white matter is made of myelinated axons. And gray matter is also less dense and it's more squishy, and white matter is more firm and dense than gray matter. The third topic that I talked about was optogenetics. Optogenetics is a research tool in neuroscience that uses a combination of LED lights and genetic engineering technologies. In optogenetics, cells are genetically engineered. Opsins are sensitive to light and can be used as light-activated ion channels or pumps. These opsins can be used to change cells' negativity or positivity, cause neural activation, silencing, stimulation, and inhibition. Opsins are optogenetic actuators. They are naturally occurring and they are light-sensitive proteins. And they can modify the activity of the cell when the cell is exposed to light. The fourth item that I talked about was Python. So Python is a programming language that is used in neuroscience. This programming language allows neuroscientists to code and analyze very large amounts of data. They can analyze neuroimaging modalities such as fMRI, EEG, and MEG. They can analyze electrophysiology recordings, and they can do this in an efficient and a fast way. Scientists can also create toolkits within Python. One of these toolkits is PsychoPy, which can generate visual stimulation protocols. Python is used for modeling and simulation environments, and it is also used in coding and analyzing large networks of neuron models and brain regions as well. The fifth item that I talked about is the difference between in vivo, ex vivo, and in vitro experimental methods. So in vivo can be translated to within the living, and in vivo experiments are carried out in living organisms such as rodents or non-human primates. And this is essential to understanding things such as behavior, emotion, and more complex models that other experiments would not be able to provide. In vitro can be translated to in the glass. These experiments are carried out on cells or bacteria, usually in culture dishes or test tubes in a controlled environment. Ex vivo can be translated to outside of the living. This form of experimentation takes living tissue from a living animal or organism, and it is cultured within a lab, and this changes the tissue very little. The sixth item that I talked about was ethics in neuroscience research. Due to our brain being connected to our identity, personality, and our understanding of who we are, it's important that we uphold ethical standards and protocols in neuroscience. There are also new and emerging technologies coming out that will allow us to dive deeper into the brain and the brain's function. And it's important to protect the individual's privacy and identity, as identity and personal autonomy are intimately linked with the brain. It's also important to establish consent that is concise, especially for those with neuropsychiatric disorders, in order to uphold these ethical procedures and protocols. The seventh item that I talked about was NeuroPixels probes. So NeuroPixels probes is an electrophysiological method that provides up to 1,000 recording sites with low noise from hundreds of neurons. This is low cost and relatively accessible to researchers. It uses CMOS technology, and it constructs silicon integrated circuits. The NeuroPixels probes has 960 recording sites and only weighs 0.3 grams, so it's very small, and it does not harm the animal when it's placed. It has the ability to isolate neuronal activity also, so it can single out a detected spike from a single neuron from over a dozen recording sites. And it can sample from multiple brain structures simultaneously, recording from multiple layers of the brain at one time. The eighth item that I talked about was comparing animal brains to human brains, particularly a sheep. So the sheep brain and human brain have many similarities, but they also have differences when looking at structures. The first item that I talked about was the spinal cord within the sheep and human brain. So the spinal cord is both attached to the brainstem, which can be found below the cerebellum. The start of the spinal cord in the sheep brain can be found posterior to the brainstem and cerebellum, pointing horizontally. In the human brain, the spinal cord can be found inferior to the brainstem and cerebellum, pointing downward. The difference is mainly in the body position, where the sheep lies horizontally and the human lies vertically. The second structure that I talked about was the corpus callosum, where the corpus callosum lies in the same location in both the sheep and the animal, or the sheep and the human brain. The corpus callosum is very thin in the sheep brain, and it's much thicker and more dense in the human brain. The ninth item that I talked about was mobile EEG. So EEG is basically a neuroimaging tool that places electrodes on the scalp and records electrical charges from brain cells in the form of brain waves. These EEGs can detect abnormalities or changes in the brain due to disorders or just presented stimuli. Electrodes in normal EEGs also have very long cables. Mobile EEG is a neuroimaging tool that can perform EEG on freely moving participants. They have a small, lightweight amplifier, and some are wireless. They also have very small cables. This differs from the normal EEG. They are wearable and self-fitting, which allows participants to freely move in a natural environment. Mobile EEG is also being used to cater to sensory, cognitive, and motor sensitivities in neurodevelopmental conditions such as autism and ADHD. For example, some people with ADHD might find it hard to sit down for long periods of time, like they would need to in a regular EEG test, but mobile EEG would allow them to freely roam around while still getting those recordings. The 10th item that I talked about was animal models in neuroscience. So although animal models have been used for many years, rodents in particular, have been used in drug and behavioral trials for neuropsychiatric disorders. One issue with animal models is that humans' neuropsychiatric disorders, such as depression, are very complex. Depression, it being heterogeneous, means that no single model is able to capture the distinct causes or mechanisms of the disorder. Rodents cannot be depressed, they cannot be schizophrenic, but they can model these symptoms of these disorders. So neuropsychiatric disorders are also usually diagnosed based off clinical criteria, not relating to brain mechanisms or behaviors. So using schizophrenia, there are clinical criteria such as delusions and hallucinations that would be quite hard to model in an animal model.